and it's my pleasure to welcome you on behalf of our team, including Bobby Jo Webb-Robertson, who is the PNNL co-director. Our speakers today are Drs. Karen Rodland and Jeff Tyner. It's a particular pleasure to introduce Karen Rodland, who was the founder and first director of PMedic. Karen served as the chief scientist for biomedical research at PNNL. Before that, she did graduate and postdoctoral training at Syracuse University and moved west to join the faculty at OHSU in what was then called the Department of Cell Biology and Anatomy. If you're counting, I think that's 2 name changes ago. She took a sabbatical in 2000 to learn proteomics at PNNL and ended up relocating, where she is focused on the application of mass spectrometry-based proteomics to improve biomarker discovery, with an emphasis on cancer and early detection. She retains an affiliate appointment as Emeritus Professor at OHSU in what is now called the Department of Cell, Developmental, and Cancer Biology. Dr. Jeff Tyner is a professor in the Department of Cell, Developmental, and Cancer Biology and is co-director of Translational Oncology in the Knight Cancer Institute. He also directs the Cancer Biology graduate program. Jeff received his PhD from WashU in St. Louis and did postdoctoral work at OHSU before joining the faculty here. His research is focused on identifying genetic lesions underlying cancer pathogenesis for individual patients and identifying specific gene-targeted therapies tailored for each patient. Today, Karen and Jeff are going to talk about the Clinical Proteogenomic Tumor Analysis Consortium. After the talk, we will have time for questions. You can type questions into the chat at any time, and I'll read them aloud at the end of the presentation. Please mute yourself if you're not a speaker, and if you hear weird thumping, it's the roofers at Karen's house, in case you have not heard about that already. Okay, take it away, Jeff.
All right, thank you so much, Beth and Bobby Jo. It's really a pleasure to be here to present, and I'm delighted to present with Karen. I'm going to try to set a foundation for the disease of AML and some of the biology around AML, and sort of the platform that we have for studying AML at OHSU. I'm going to try to be efficient in the first part of this presentation because the second part, where Karen will present, has really all of the proteomics and the collaboration with PNNL, and that's really the exciting part. And that, I should say, was born from a U01 award as part of the CPTAC Clinical Proteomic Tumor Analysis Consortium. It's a very large consortium. Karen will tell you more about it. She has done an outstanding job over the past 5 years being the contact PI for this U01 CPTAC PTRC, that's an alphabet soup, Proteogenomic Translational Research Center, which is the collaboration between OHSU and PNNL that has led to all this work. And should be noted, we just put our competitive renewal in over the summer, so fingers crossed that we get well reviewed and have another 5 years. So the subject matter of this project and this collaboration is acute myeloid leukemia. And this is obviously a blood-borne cancer, a heme malignancy. It is one of the most common forms of heme malignancy. There's about 20,000 diagnoses every year in the United States. The standard therapeutics, by and large, for decades have not changed, and they combine, for the most part, anthracyclines with nucleoside analogs. That's the most commonly used 7+3, 3+7, different names for it, approach. There are subsets of disease for which alternative therapies are used, probably the best example being the ATRA therapy used for acute promyelocytic leukemia, which really has become sort of a distinct disease entity unto itself. By and large, unfortunately, with the exception, the real exception of this ATRA therapy, which works very well, by and large, the other therapeutic approaches have not worked well, unfortunately. And this is just shown over many decades on this survival plot on the left side of this slide, where we can see that since the 70s, there's been virtually no improvement in outcomes for AML patients using these anthracyclines plus nucleoside analog approaches. And so this has sparked a real interest in developing, of course, new, better therapies for AML. And like so many other cancers, there's a real interest in applying precision oncology, which oftentimes looks something like what's shown on the right side of the slide, where a patient may be diagnosed with AML, and there's some sort of omic analysis that drives the decision to use alternative treatments, novel treatments, combination treatments that lead to better outcomes. That's how the story is told. That's how we would like it to be. And on the surface, you could say there has been some progress in this regard. Over the past 5 years, after decades without any approvals, there have been, I think, 7 or 8 new approvals for AML, FDA approvals over these last 5 years or so. I've summarized several of these on this slide. In several cases, there have been approvals for small molecules targeting enzymes that are mutated in AML. FLT3, you're going to hear more about from Karen, is the most commonly mutated gene in AML. It's a kinase. These are gain-of-function mutations, and so FLT3 kinase inhibitors have been approved. IDH1/2, also mutated enzymes for which there are selective inhibitors. And then there's some other approaches. I'll talk a little bit about BCL2 inhibitors, which have shown some promise in AML. And there's a lot of other strategies being explored. And so on the surface, this sounds great. There's all these new FDA approvals. And if you look at the initial response rates, it does seem encouraging that there are some good responses initially to some of these new therapeutics. Unfortunately, these are almost across the board not durable when used as single agents, or even in some cases when used in combination with other agents. Almost all patients still, after some time, experience drug resistance, disease relapse, and so we still have a lot of work to go. Why is it that these targeted therapies based on rationally guided, genetic guided approaches are not working more durably? To understand that, we have to look a little deeper into the biology of this disease. There's a number of papers that have looked sort of pan-cancer analysis, pan-genomic cancer analysis, and looked at the mutational burden of different tumors. And every time in all of these papers, acute myeloid leukemia, AML, is always on the quiet side of the landscape, meaning that there are fewer mutations per tumor in AML than in many other tumors. And so this, I think, this is absolutely true. It's absolutely accurate data, but I think it gives a little bit of a false impression that this is a genetically simple disease. When we look at the data across tumors, it becomes much clearer that this is a very genetically complex disease. And this is from the Cancer Genome Atlas data for AML, published in 2013, 200 AML patients, which are shown on this matrix plot, every column being one of those patients. Every row being a gene or a family of genes. And we can see that there's, across 200 patients, really incredible genetic diversity. Even though there may be only somewhere around 5 to 15 mutations per tumor, they occur in many different combinatorial patterns, and almost every patient shows a different mutational profile. And this can be shown even in greater detail when looking at just 7 of the commonly mutated genes in AML on this pie chart. Leads to something around 40 or 50 different combinatorial mutational patterns that can be observed across different patients with this disease. In addition to the genetics, which I think clearly are maybe more complex than might be inferred through some sources, there's also additional factors that complicate AML history and therapy. About 20 to 30% of AMLs arise either from a prior solid tumor, or a prior form of cancer which a person received a DNA damaging therapy for, and then some years later developed AML as a result of the DNA damage of that previous round of therapy. Those are called therapy related. Another 10 to 20% are secondary to a prior myelodysplastic syndrome. And these are very complex, genetically complex cases that are very recalcitrant in many cases, most cases, to therapy. And so those are very difficult to deal with. We also have, as I'll talk about in just a minute, the cell biology of the disease, the cell differentiation state of the blasts, the AML blasts, which range all the way from completely undifferentiated cells all the way through to really pretty mature looking cells, monocytic, basophilic, erythroid looking cells. This usage of morphology to classify AML was very popular 10, 20 years ago. It has really been replaced with genetics of late, but I'm going to make an argument before I hand it over to Karen that we perhaps need to explore or reincorporate these morphologic states, which I think may explain responses and resistance to some of these newer therapies and possibly to some of the older ones as well. So I think, to summarize some of the challenges that we have with precision medicine and AML, it seems very logical. You just take a mutational profile and diagnose a drug based on that profile. But we have some really important challenges in that there's a lot of genetic heterogeneity, as I tried to illustrate. Many of the mutant genes we don't actually have drugs for. In many cases, they're loss of function mutations. We don't understand the biology well enough yet to understand what the drug that should be used for those loss of function mutations. We have this whole issue of cell differentiation state, which is not yet explored fully. And then finally, the genetic signature of AML tells us really nothing at this point about the microenvironmental factors or immune suppression that may also be playing a role in the disease. And so some years ago, I think at this point, about 7 or 8 years ago, we established the BEAT AML program. And this was an effort that was a coordination with the Leukemia Lymphoma Society and the Knight Cancer Institute, as well as about 11 or 12 academic medical centers around the country. And OHSU was sort of the central component of this, where we received samples from all of these dozen or so medical centers, processed these samples. The goal was to develop a cohort of about 900 AML patient samples. We've surpassed that at this point. Perform functional screens, and what that means, I think I have on the next slide, is essentially ex vivo drug sensitivity. I'll talk about that in a second. Perform exome and RNA sequencing on a subset, study the immune microenvironment, which we won't have too much time to talk about today, and then through computational biology, integrate all of these data together to nominate biomarkers or mechanisms that can explain drug sensitivity or resistance and parlay that information into clinical trials. And so we have completed, in a sense, the first phase of this program, in that we did collect actually more than 900 samples. And the first sort of tranche of data from this data set was published in Nature, hard to believe, almost 3 years ago now. And I'll just walk you through sort of what's in that paper very briefly, just to give you a little more background on the ex vivo drug sensitivity assay. This is really, in many ways, a pretty simple concept. It's just high throughput screening, except rather than using cell lines, we're using primary patient material. And so we can take blood or bone marrow from AML patients, isolate mononuclear cells, whole mononuclear cell fractions. We process the samples immediately. We have sort of an ongoing staff that's constantly on call to receive these samples, process them, and then immediately plate them against libraries of drugs. And then 3 days later, perform a colorimetric high-throughput cell viability assay to understand patient-specific drug sensitivity profiles. And so we have done this now, a lot of this. It's, again, over 900. In fact, if we look across all the heme malignancies we've screened over the past 10 to 15 years, we've screened greater than 2,500 heme malignancy patient samples. And we really see this as a visual that shows the small molecule drug sensitivity results. And you can see that there's really a lot of heterogeneity of drug sensitivity across these patient cohorts. Looking, I think, in this case at about maybe 300 or 400 patient samples screened against about 125 different drugs. We can also see that this heterogeneity of drug sensitivity matches a similar plot to what I showed you earlier from TCGA. This is our version from BEAT AML, where we actually have now an even larger cohort than TCGA, and we see similar patterns of genetic heterogeneity across the patient cohorts. In fact, if we compare BEAT AML to TCGA, we do see at the most common side of mutations, those that are most commonly observed, we see pretty good lineup of the mutational frequencies, some notable outliers like SRSF2, but by and large, the most commonly mutated genes, we see very good agreement. If we look at the long tail, the very rarely mutated genes, there's actually a lot of disconnect where we are seeing mutations that were missed in TCGA. Conversely, TCGA found mutations that we've not seen. And so I think even with collectively over 1,000 patients sequenced between the two efforts, we still probably haven't fully captured the genetic heterogeneity of the disease accounting for all those private mutations. From a transcriptional profile, we've also performed RNA-Seq on all of these samples. And we see that, again, there's a lot of diversity of gene expression patterns, some of which do tend to organize a little bit around some of these recurrent genetic features, such as mutation of NPM1 or some of the rearrangements that are observed in AML. We did include healthy bone marrow, either whole mononuclear cells or sort of primitive CD34 positive enriched bone marrow cells as comparators for the AMLs in this RNA-Seq data set. We did our informatics computational team led by Shannon McQueenie did just an incredible job with every aspect of this project. I should mention they're still doing an incredible job. We actually have a new tranche of data that we're readying for a new publication, and which we hope to be submitting anytime now, probably in the next couple of months. We'll be submitting a new paper that releases a whole new set of data. But the data that's currently been released in that original paper is all available in many different places, one of which is this home-built web portal called Visome that Shannon and her team did just an incredible job of developing a point-and-click tool that I think leads to a democratization of this data set, where you don't have to have any programming skills to be able to go in and ask questions, visualize, explore the data set. And this website has been extremely well accessed and had and I think in the first month or so, it had 10 or 20,000 hits on this website. So it's been a very popular tool for people to explore this data set. So, what can we do with these data? It's one thing to show the landscape of mutations, gene expression, drug sensitivity. What we really want to do is to integrate the data. And so that's really what that first Nature paper did, was to make predictions about, for instance, if you have mutations in particular genes, those can confer either greater sensitivity or greater resistance to one or more of the drugs on the panel. We could look at the gene expression data, and we could pick a drug and say, what are the 20% most or least sensitive samples to that drug? Can we find a gene expression differential expression signature that distinguishes those most from least sensitive samples? And the answer was, for about 2/3 of the drugs on the panel, we could find a statistically significant signal. We could be even more nuanced with the approach and look at the gene expression and drug sensitivity both as continuous variables, overlaying also mutations on this very complex looking buzzsaw plot, as we call it. And this approach allows us to extract additional signal regarding correlations between drug sensitivity and mutations versus expression that were not observed in the more simple approaches above. And then finally, we could look at we really spent a lot of time collecting detailed clinical annotations. Nicola Long on our team is our clinical data manager and does just an outstanding job of collecting accurate, detailed clinical annotations on this whole patient cohort. And so we have the opportunity to understand that, for instance, although you can't read it, this is looking at some of those de novo versus the MDS transformed AML cases and seeing that we in fact do see some differential drug sensitivity between those clinically distinct subsets. So long story short, this paper produced many predictions of biomarkers for drug activity, almost all of which, or many of which, we still are working on developing mechanistic and clinical validation. And so with the last couple of slides before I hand it over, I want to show you just a couple of examples of how we have leveraged into some newer studies where we have been able to do just that. And before I get to those, I'll just mention that, to our knowledge, this is the largest functional genomic data set of primary tumors that has been produced to date. And that we do feel that both the public access of the data as well as just leveraging of this large data set can really help to inform new biomarkers and drug combinations. And so I think, in essence, although the first phase of the project is done, it's really just getting started, and in particular, the fun part is just getting started. So just a couple of really short vignettes before I hand it over to Karen about how we have been able to leverage this data set. And I'll begin with one of these newly approved. And in fact, I think this may be the most recent approval for AML, is the BCL2 inhibitor Venetoclax, which is used in combination with hypomethylating agents for elderly AML patients who are not fit for the current standard of care cytarabine plus daunorubicin regimen. And as I mentioned earlier, the initial remission rates seemed incredible, and this is really a difficult to treat population. And the initial remission rates of 60 to 80% are just far better than anything we've seen before. Unfortunately, this has not held up durably, and so after 1 year, that response rate goes down to 30 to 40%. And I think almost all patients who have seen this eventually have relapsed, unfortunately. So it's not been a long-term solution. And so we wanted to understand what are the mechanisms of resistance and potentially biomarkers of sensitivity to this BCL2 inhibitor. And so we turned to the data set and asked the question, what does correlate with Venetoclax sensitivity and resistance? And we've just recently published this, may have actually been a year ago at this point, published this paper in Nature Cancer. And what we found was that one of the most striking correlations was in fact not necessarily genetic, but it was those cell states, where we found that the AML tumors that had a more monocytic differentiation state were more resistant to venetoclax. Those that were more primitive were more sensitive. This was a surprise because typically speaking, those more primitive cells have been described as the, quote, leukemia stem cells that everyone, feels are problematic and give rise to resistance, at least in response to the standard chemotherapy approaches. We found that venetoclax tended to show the opposite. It does correlate to some extent with mutational profiles where there are particular gene rearrangements, in particular that PML-RR alpha, which is extraordinarily sensitive, and these do have very high levels of BCL2 expression. And then we did see that there were a smattering of point mutations, particularly in P53, RAS, SF3B1. Some of these do correlate with monocytic differentiation state that were resistant, so that is very consistent. And then we could turn to the gene expression data and understand that some of the BCL2 family members, alternative usage of BCL2 family members such as BCL2A1 was correlating with extreme resistance to venetoclax. If one looks at normal hematopoietic cells, one can observe that in the monocytic state, we do see downregulation of BCL2, upregulation of other BCL2 family members, such as BCL2A1. So I think that all of these are interconnected, and in fact can be shown on this little cartoon diagram where we can see that cells of a monocytic state, sometimes which might be driven by mutations in RAS pathway or P53, have altered utilization and expression of BCL2 family members, altered mitochondrial metabolic states, which correlates with resistance to venetoclax. And comfortingly, this has been confirmed. These findings have been also seen by several other groups around the world, groups both in Finland and in Denver using sometimes similar approaches, but completely orthogonal data sets, have made these very same observations that venetoclax is less effective against monocytic myeloid cells. And in fact, the Denver paper in Cancer Discovery showed that in patients clinically treated with venetoclax, relapse was often observed with a monocytic phenotype of the cells that were relapsing. Similarly, in Montreal, a group using a similar approach but distinct cohort of samples observed BCL2A1 to be one of the strongest correlates of resistance to venetoclax. And then finally, the clinical trial data shows us at the genetic level that P53 and RAS pathway almost always correlate with either primary refractory or disease relapse. So it's very nice to see that many groups around the world are coming to the same findings around venetoclax sensitivity and resistance. And just finally, one similar finding, except the opposite of what I just told you, we just very recently, Kyle Romine, a graduate student that I co-mentor with Evan Lind, has put out a paper in Blood Cancer Discovery, again leveraging this Beat AML data set as well as some genome-wide CRISPR screening to study mechanisms of sensitivity and resistance to BET inhibitors, BRD4 inhibitors, and finding really the converse story of what I just told you with venetoclax, that in contrast to venetoclax, which is more effective in the primitive, these BET inhibitors seem to be more effective on the monocytic, and resistance seems to correlate with those more primitive cells. And so consequently, combination of BCL2 plus BET inhibitors is a very attractive option to us at this point. So, long story short, we have developed this very large data set. We have been able to use it to leverage some interesting findings regarding drug sensitivity or resistance. That said, we've been missing, until this collaboration with PNNL came along, we've been missing an obviously very important component of data to this entire data set, which is proteomics and phosphoproteomics. And so we were thrilled to jump at the chance to collaborate with Karen and the team at PNNL to apply mass spec-based proteomics and phosphoproteomics to this cohort, and adding this new data dimension, as you're going to see from Karen, has really led to some important and impactful findings that are already being leveraged into sort of new biological understanding and into some forthcoming clinical trials. And so with that, I'm going to hand it over to Karen. And Karen, I'm happy to advance the slides for you. And you can just say next slide, and I'll push the next one.
Please do that, because I can't guarantee anything technical at my end. So this is where the collaboration between OHSU and PNNL came in, and PNNL has been funded in several prior iterations of the Clinical Proteomic Tumor Analysis Consortium, which was really initially focused on complementing TCGA analysis of multiple tumor types with in-depth quantitative global proteomics and adding phosphoproteomics onto measurement of protein abundance, but also measuring phosphorylation at specific peptides. And in 2015, they came out with a call for the Proteogenomic Translational Research Centers to try to leverage the NCI-funded clinical trials and get proteomics applied to clinical trial samples. So all the PTRC sample centers were supposed to have both a preclinical arm, which was our kind of standard proteomics with retrospectively collected samples, with clinical outcomes data, or preclinical models like cell lines and PDXs, and derive a signature for something relevant, translationally relevant. And at that point in time, we were picking the ability to predict the response to the small molecule inhibitors. And we were also intended to have a clinical arm using actual clinical trial samples to validate the conclusions that came from the preclinical arm. And the little US map just shows where the various centers are. It really is a national consortium. Next slide, please, Jeff. So this is the PNNL OHSU PTRC and our initial specific aims. In the preclinical arm, we wanted to leverage the beautiful ex vivo drug response data that Jeff and colleagues had come up with that was so key to the Nature paper, and see if we could develop a proteomic signature based either on protein abundance, which we call global proteomics, or protein phosphorylation, which we call phosphoproteomics, to predict the response of a particular patient in the ex vivo IC50 assay. Then in the clinical arm, we wanted to leverage Beat AML samples, as well as a couple of small clinical trials at OHSU, to validate the signature and see if we could reproduce in clinical samples with the proteomic signature the ability to predict the ex vivo signature, and then if we could, to actually compare the two predictors, the ex vivo response and the proteomic data. And I have to say that OHSU has always been PNNL's go-to place for our clinical partners. We've got some of the best mass specs in the world and people who know how to use them, but they're no good unless you have an interesting clinical problem to tackle. And that's what we got from collaborating with Brian and Jeff and OHSU. So if I can have the next slide, please. So our preclinical experiments, we started with cell lines. A lot of this was to work out the methods. And so there are 4 AML cell lines that are incorporated into the ex vivo assay panel by the FDA so that you get a known response to benchmark. And these are K562, which are BCR-able cells, MOM14s with a FLT3 ITD mutation, HL60s with an NRAS Q61L, and CMK with a JAK3 mutation. And we treated each one of these cell lines with the appropriate inhibitor for its driving mutation. Dasatinib for K562, sorafenib for MOM14, trametinib for HL60, and ruxolitinib for JAK3. And we also did a time course study. If I can have the next slide. So the time course study was 0 times, 30 minutes, 3 hours, and 16 hours. Look at this as a mega Western. Instead of being limited to the proteins and phosphoproteins that we had antibodies for, we could look at everything all at once and use computation to sort out the 50 most differentially abundant or most differentially phosphorylated things. And we're no longer looking under the lamppost. We're looking at everything and trying to assess its change. And from just the rough heat map that you can see on this slide, you can see that there are varying patterns of response. Sometimes things change quickly, sometimes they change slowly, sometimes they revert to where they were at 16 hours after 3 hours. But if I can have the next slide, please. Working with Amick and Ozgun at OHSU using their causal path tool, we were able to identify the pathways that were most differentially affected, not just the individual proteins from that heat map, but the pathways, which I always am convinced contain the most significant information about how the cell phenotype is changing. And the pathways are the executioners of the driving mutations. And that is how you can access the ways in which multiple mutations in the same cell line interact to modulate the phenotype of the cell. So some of our conclusions from this very preliminary pilot study actually, was that in both the MOM14s and the HL60s, we saw the first effect of the inhibitors was in the MAP kinase pathway, which was quickly downregulated at 15 minutes and 30 minutes. And this is interesting, even though there are two different driving mutations. You have the FLT3 driving mutation versus a RAS, but think about it, FLT3 feeds into the RAS pathway. It's not that unusual that the downstream events should converge, which is one of the driving motivations behind this proteomic study. Can we find the converging parts on pathways that are treated with multiple inhibitors or have multiple driving mutations so that we can actually target a point of convergence that has multiple upstream modulators and have a more robust patient response? For the CMK with the JAK mutation, we got a very rapid and nice, almost textbook downregulation of the STAT pathways. In general, the 3-hour time point looked a lot like the 30-minute time point, just more so. But consistently in all 4 cell lines, after about 16 hours, we saw upregulation of apoptotic pathways. So these cell lines that we were known to be sensitive to these inhibitors were starting to die. We went on to build a combinatorial experiment, which I'm not going to report on because we haven't analyzed the data yet. May I have the next slide? Because that leads to what the real challenge is. And the real challenge is, how do you interpret all these data when you have RNA, DNA, proteome, and phosphoproteome from the same patient? So we started by selecting a cohort from the Beat AML-1 study of 38 patients, all of whom had the ex vivo drug response and clinical annotations, proteomics, RNA-Seq, and whole exome sequencing. So this is just a list of the 38 patient IDs that we selected. We handed this over to our bright computational people, Sarah Gosling predominantly, to come up with a workflow for integrating mutations, transcripts, proteins, and phosphosites across patients in the context of their drug response. May I have the next slide, please? So the first line tool is our correlation tools, correlative tools. What mutations correlate with what drug response? What changes in protein abundance correlate with what changes in drug response? And we basically used 2 different methods of doing linear regression, of doing this comparison. One is a lasso regression technique, and you can think of it as a continuous versus a continuous technique. We take a look at the level of abundance of genes, transcripts, proteins, phosphosites, and correlate them with the area under the curve measured in the ex vivo drug response. So it's a linear to linear. And we used leave one out cross-validation to measure the mean squared error. So that is the difference between the observed drug sensitivity and the predicted drug sensitivity. For the logistic regression approach, we binarized the population. We either defined a patient as being sensitive to the drug or resistant to the drug. And we used an area under the curve of 100. If it was 100 or less, they were sensitive. If it was greater than 100, they were resistant. This can kind of crispen up the data by getting rid of the middle ground where they're more or less resistant. And so, again, we were using this approach to identify features that predict being classified as sensitive or resistant. And so we were looking at misclassification error. Next slide, please. Both approaches worked quite well, but slightly differently. So when we use the LASSO approach, as you can tell in this slide, and hopefully you can see my little cursor, the gray boxes on the top row are the sensitive patients, and they correlate with light blue areas under the curve in the second row of ribbons, and the white are resistant, and they have the darker colored area under the curves, the higher values of area in the curves. And we can see that sensitive and resistant patients stratify quite nicely. There are a number of different proteins that appear to be driving the clustering. And we see the same thing with phosphocytes. So the smaller panel below looks at a particular peptide that has a phosphorylation on a particular amino acid, serine 1218 in MAP18, for example. So this was using quizartinib response, which is a FLT3 inhibitor, and we got some very nice data. Nice. Next slide, please, Jeff. We then used the logistic prediction to compare the two methods and workflows, and same footnotes here. Gray boxes are sensitive, white boxes are resistant, and we got tighter clusters. We got some very strong apparent correlations, which does happen when you do this. And then, but there was very little overlap between the apparent driver proteins between the first method, the LASSO method, and the second method, the logistic method. So we need to go in and figure that out. One can take this simple approach of saying those things that do replicate in both methods are more robust, but we have to do some actual looking at pathway members to try to figure out why the overlap is not tighter than it is. This work has been written up, and we're publishing it as a way to certify the workflow that we're going to use with a 220 patient cohort derived from the Beat AML 900 patient cohort, and where we're basically going to correlate the protein signatures with the ex vivo drug response. Next slide, please. So a summary of what we found in this preliminary workflow was that both proteomics and phosphoproteomics did a good job of distinguishing drug sensitivity, even with a very small sample cohort. So we have a cohort of 38 patients, and we're measuring thousands of phosphopeptides and 10 to 15,000 proteins. So that's a model for what we call false prediction or overfitting, but we were able to get some really reasonable data despite the small sample cohort. When we compared the error measurements with the proteomic data compared to the error measurements with DNA mutations or transcriptomic data, we actually found that the proteomic model was more robust and had lower error values than either DNA mutations or RNA-Seq expression. They weren't hugely different, but they were consistently better when we looked at the protein values as compared to mutations or RNA-Seq. And so we're revising and going on with the larger cohort. So that was what we had set out to say we were going to do. And as always happens with good science, as you get doing it, you find an even more interesting question that is worthy of your effort. So if I could have the next slide, Jeff. One of the major targets in Beat AML and in AML is the FLT3 receptor. And FLT3 inhibitors have become quite successful in the clinic. A third of all AML cases have mutations in FLT3, either an internal tandem duplication that is in frame or point mutations that are activating mutations of kinase activity. Either type of mutation makes a constitutively active FLT3 and stimulates cell growth. There have been multiple FLT3 inhibitors developed. Some of them are dirty and have a lot of cross-reactivity. Some of them are cleaner. Many of them have a very nice initial response, but almost all of them demonstrate the eventual development of drug resistance. For this study, we focused on gilteritinib, one of the more recently FDA-approved inhibitors. I think it was approved last February, just before the COVID shutdown. And it showed a really nice 6 to 7 month initial response. May I have the next slide, Jeff? So this is work of Ailey Traer and Sunil Joshi, and they had the brilliant idea, to me, of looking at the development of resistance over time, not just the late resistance that you have after growing a cell line in the presence of inhibitor for months and months and months, which the field has tended to concentrate on, but to look at the process of selecting from initially sensitive cells to resistant cells and what are the intermediate stages. And so this is what actually happens in the patient during the early days of treatment when you have your initial response, and it happens in the bone marrow. So they theorized that factors in the bone marrow could be contributing to the survival of the AML cells during this early resistance stage. So could I have the next slide, please? So they started out by culturing primary patient, the cell lines, the MV11, MV411, and the MOM14 cell lines, in the presence of gilteritinib, which is the yellow line, and the cells did not grow, or in the presence of gilteritinib plus either FGF in teal or FLT3 ligand in the reddish color. And they could see after a certain amount of time, somewhere around 25 days, the cells that were cultured in the presence of ligand started to grow out again. Next slide, please. They termed this early resistance. No, stay right there. Then if you took these early resistant cells and removed the ligand, you took the FGF or the FLT ligand away, they crashed, they died, but after a certain length of time, a new population grew out. And they termed this ligand-independent culturing or late resistance. May I have the next slide now? And their concept was to characterize all aspects of the transition from early resistance to late resistance in a very multi-dimensional study. They used whole exome sequencing, small molecule inhibitor sequencing, proteomics and phosphoproteomics, metabolomic analysis, and then hypotheses that came from the descriptive data were validated with a certain level of CRISPR-Cas9 screening. This work was just recently published in May in Cancer Cell. This was just the most fun project I've ever had. Next slide, please. So if you take a look at NRAS mutations, which are very prevalent in the late resistance environment, you see both the MOM14 and the MV411s had large numbers of NRAS mutations, although they were different specific mutations. You see that in very low levels of FLT3 mutations. And this matches the clinical data. This is known to be what you find in patients. May I have the next slide, please? But if you take a look at the emergence of these NRAS mutations, you find that initially during ligand-dependent culturing, when the cells are beginning to grow out, they still have a very low allelic frequency of mutant RAS. It is not until well into the ligand-independent culturing that you really see a massive outgrowth of the RAS clones. So the NRAS mutations were not necessary for the ligand-dependent early resistance phase, and this was confirmed with CRISPR-Cas knockdown experiments. May I have the next slide, please? Looking at the metabolomics during this transition was also very interesting. The parental cells, as we know in culture, depend on glucose metabolism. But as you switched them to early resistance, you saw a decline in the products of glucose metabolism and a gradual increase in non-glucose related sources of energy, particularly lipid metabolism, and interestingly, two different classes of lipids in the two different ligands. The FGF2 late cells had upregulation of sphingolipids, and the FLT ligand had carnitine and fatty acid metabolism. Next slide, please. And this is where the PNNL collaboration came in. We were asked to do the proteomics and phosphoproteomics, and the very initial data analysis that we do when we do these kinds of studies is a PCA analysis to see who groups with whom. And we did both global and phosphoproteomic, and initially it was so striking. The parental cells all grouped by themselves, the ligand-dependent cells grouped further away from the parental cells, and the ligand-independent cells all grouped by themselves. And as you can see again in the teal for the FGF2 and in the red for the FLT3, the two different ligand classes also grouped together within this larger group of early resistance. And this held even into late resistance. And this clustering was even more prominent with the phospho data. And this really led us to think that we might have a key with the proteomic data to understanding the actual process of transition from early to late resistance. May I have the next slide, please? So we did kinase enrichment, kinase substrate enrichment analysis, which counts the phosphorylation level on known substrates of a kinase and gives a score to kinase activity based on the percentage of known substrates that show increased phosphorylation as a result of your perturbation. And we found some very interesting differences as we went from early resistance to late resistance. In the ligand-dependent culturing, the early resistance, basically we saw a slowing down of cell cycle proteins. Multiple CDKs were downregulated in the early resistance compared to the parental cells. But there was a very marked and significant increase in aurora kinase B signaling, which was then lost when we went to the late resistance. We found upregulation of multiple cell cycle related proteins, indicating recovery of cell cycle function. We saw a hyperactivation of MAP kinase pathway members, which would go along with the NRAS activation. And we had lost that nice increased aurora kinase B. So next slide, please. That led to the hypothesis that perhaps if we inhibited aurora kinase B during that early resistance phase, when we were doing the ligand-dependent culturing with FGF or FLT ligand, which, if you remember, is supposed to be mimicking the stromal contribution to survival during early resistance, could we perhaps prevent the development of late resistance or have a more complete response in early resistance? And you can see from this slide where we treated in black with gilteritinib alone, in blue with a novel aurora kinase B inhibitor, AZD2811, and the combination of the two. When we added the aurora kinase B inhibitor, we left shifted the viability curve, and we got cell death much earlier, and we mitigated the ability of the ligands to promote survival in the early resistance phase. It was not quite as effective looking at the late resistance phase. The combination did have some effect, but not the telisolizumab by itself. And this also does kind of make sense if we think that the aurora kinase B is really only contributing to the early resistance phase. Now, these were cell line studies. May I have the next slide, please? What happens in patients? And this is, so we, Amy Traer had a gilteritinib clinical trial going on, and timing-wise, this was just at the start of COVID, but because it was a treatment human subjects protocol, it got to continue. So we actually got to continue this work. And so the thought was that the early resistance phase in vitro is modeled by the early response phase. So we had pre-treatment samples from FLT3 ITD patients who were then treated with the FLT3 inhibitors, and a bone marrow sample was taken at day 28 to 56. And we either had sensitive or resistant patients at that stage, and relapse, the return of very large tumors, would be analogous to the late resistance phase, whereas these cells that survived the initial treatment would be the early resistance cells. And again, they separated out very nicely on PC2 when we took a look at a heat map of the kinases that were up and down regulated in the pretreatment and the early resistance phase. They separated out very nicely. And when we did the pathway analysis on these results, it was very, very consistent with what we had seen in vitro with the cell lines. We saw a decrease in cell cycle related proteins. We saw an increase in MAP kinase related signaling, and we saw an increase in fatty acid metabolism. And so based on this work, there's actually a clinical trial that LA is the PI on, being proposed to combine the Aurora kinase B inhibitor with the FLT3 inhibitors in actual patients. May I have the next slide, please? This is the actual data on drug sensitivity in samples from 4 patients. With one exception, we got a very nice response during the early resistance to the gilteritinib plus the Aurora kinase B inhibitor. In general, at pre-treatment, it didn't make a difference. There was no difference in cell viability from the Aurora kinase B inhibitor or the gilteritinib or the combination with, like I said, one patient. But in all 4 patients, the combination really did decrease cell viability. So potential therapeutic approach, motivation for a new clinical trial. To me, this is a beautiful series from going from very basic science, what are the mutations, what are the changes in RNA expression, protein phosphorylation, to actually a mechanistic explanation, which, by the way, has also been tested by CRISPR-Cas knockdowns and knock-ins, and leading to a potential translational output. And because of the collaboration with OHSU, because of the great reputation of OHSU for translational work, we can go very smoothly into the patient and actually test this out in a clinical trial. So if I may have the next slide, please. So the model that has come out of this, and there are similar parallel data with quizartinib, which is an earlier a type 2 inhibitor of FLT3. So it specifically targets the kinase activity in the point mutants, whereas gilteritinib targets the FLT3 ITD mutations. And but we saw the same pattern with quizartinib as with gilteritinib, that during early resistance, we saw a downregulation of cell cycle, and we saw CDC7 being essential for the quizartinib early resistance, and Aurora kinase B being essential for the gilteritinib resistance, and then the outgrowth of the mutations that conveyed late resistance. So if I may have the last slide, please. So proteomics is a team sport. Translational research is a team sport. We have pictures of the PNNL integrated omics team and pictures of the Beat AML team, and all of the specific contributors marked down here. The early and late resistance work was really the PhD thesis of Sunil Yoshi under the guidance of Ailey Traer, and has been presented in much greater detail with much more flair for the biology of the FLT3 inhibitors and the FLT3 kinase by Joshi during his thesis defense. So I want to thank you, and we have some time for questions. We'll be glad to take them. And my roofers were quiet.
Thank you, Jeff and Karen. That was really wonderful work. Very fun to hear that story. I've heard Sunil's version, and it's nice to hear your version. We have a little bit of time for questions. If people have questions.
Hi, Karen. This is Sanjay.
Go ahead, Sanjay.
Excellent work. I'm curious, you showed last few slides with quizartinib and FLT3, and from the resistance point of view, you know, when you see in vivo and in vitro analysis, and when you use this combination of, I think it was AZD2811 and quizartinib, you see that a reduction in the growth. So is it fair to believe that the FLT3 is the target for quizartinib?
I should defer that question to Jeff.
Yeah, I can take that, Sanjay. That's a great question. And in fact, one of the papers that was referenced on that slide was from Neil Shaw's lab down at UCSF. And the point that was a Nature paper, the point made in that paper is that the clinical response to quizartinib actually confirms that FLT3 is the target, because the resistance mutations that are seen with quizartinib are in fact the activation loop mutations in FLT3 itself. One of the big differences, as Karen mentioned, between these two inhibitors is that while gilteritinib and crenolinib have the ability to block those activation loop mutations, quizartinib doesn't. And so what we see is, whereas you have RAS pathway mutations selected for in the context of gilteritinib, you actually see FLT3 point mutations selected for with quizartinib, similar to the way that you see ABL mutations selected for with ABL kinase inhibitors in CML. So I think we can say that yes, based on the quizartinib data, that FLT3 is almost certainly an important target. Whether there's others that are important too, that could be.
All right. So you are your last line would have been my next question. So do we know what is the FLT3 is talking to? What is the next door neighbor that influences the expression of FLT3?
Yes. So I think that's the strength of the proteomic data and the phosphoproteomic data, to be able to draw out those pathways and to identify side targets in addition to the primary target. And Jeff would have the answer as to what is known about what's upstream and downstream from FLT3. But the whole concept of doing the causal path type analysis on the phosphoproteomic data is to tease out relationships between pathways that were not evident before that. And now I'll shut up.
No, where I was going with this was because if we know the other potential another protein which is influencing the expression of FLT3, if we can have a combination attack on both sides, then probably maybe at a lower dose you can have a synergistic effect maybe. So that's just a thought.
Yeah, it's a great thought. I mean, and I don't know that we do know the answer to that. We certainly know a lot about what happens downstream of FLT3 signaling, STAT activation, MAP kinase activation, et cetera. But as far as the pathways upstream, the transcription factors upstream, there may be some literature, but it's not well established, I don't think. It's a good question.
Nonetheless, great work. Thank you.
Thank you. Again, by looking at baseline levels of FLT3 expression and attempting to correlate that with responsiveness to the drug, one can get at some of the questions is there are inherent differences among patients in the number of FLT3 inhibitors that they start out with, or does that change in time? But I will say this, that to date, when we look at the proteomic data, we do not find abundance of the inhibitor target itself as the major mechanism that we're looking at. We see very little changes in the abundance of the target. We will see changes in the phosphorylation of the target. We will see changes in the abundance of downstream effectors of the target. But we very rarely at the proteomic level have seen changes in the abundance of the target itself.
So sorry, that if it's okay, Beth, can I ask another question?
Yes, and I don't see any other questions coming up in the chat. So you get one more question, and then we're going to have to wrap up.
Thank you. So I'm sorry if I don't remember exactly what the data what you had shown was. Is it quizartinib alone, or is it in combination with 2811, AZT2811, that you see the more effect? What I'm thinking is, if one was to develop a selective FLT3 inhibitor, which not knowing the structure, information on that, that probably be attractive in terms of the therapy rather than having to do a combination to go, indirect route.
Yeah, I can take that one. Absolutely, Sanjay, that would be of great interest. It was the combination in that early resistance stage that was effective. And a more selective FLT3 inhibitor would be extremely attractive, not the least because quizartinib itself actually has really struggled, and I think is probably no longer on a path to clinical development because of side effects, QT prolongations that were observed in clinical trials, which are almost certainly due to some of the off-target effects of that inhibitor. So yes, a more selective, cleaner FLT3 inhibitor would be of great interest to the.
Great. So I think we should have a separate talk. We may have something for you to start with.
Yep.
Thank you.
All right. Well, that's a productive post-seminar discussion right there. I want to thank Jeff and Karen again. And we're past 4 o'clock, and I know a lot of people have other things happening. So we're gonna we're going to wrap this. Thanks a lot for joining us, and we'll see you next quarter for another PMedic seminar. Thanks.
Thank you for inviting us. It was a great.
Thank you.
Oh, it was really, really fun.
Thank you.